Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
We always hear about how the blood-brain barrier is like our brain's super strong fortress, right? But what if I told you that with age, that fortress starts to kind of weaken?
Yeah, and even worse, in neurodegenerative diseases like Alzheimer's, that weakening it really speeds up.
Wow, so what's happening like at the cellular level? What's causing this breakdown?
Well, that's a great question, and a new study in Nature Neuroscience might have some clues. They focused on a protein. It's called TDP-43, and this protein seems super important for keeping the blood-brain barrier, well, intact.
TDP-43, huh? Okay, I'm not familiar with that one. Tell me more.
So TDP-43, it's a protein that normally hangs out in the nucleus of our cells. And it has a really important job there. It helps regulate gene expression. But here's the thing. In some neurodegenerative diseases, TDP-43 kind of goes rogue. It clumps together and basically starts messing up all these important cellular processes.
So this study was looking at how TDP-43 might be involved in, the blood-brain barrier getting worse.
Exactly. They looked at brain tissue from 92 donors, some with neurodegenerative diseases and some without. They wanted to see if there were any patterns or differences in, the levels of TDP-43. And of course, they wanted to see how that affected the blood-brain barrier.
So did they find anything? Was TDP-43 acting up in the brains of the people who had those diseases?
Oh, the results were pretty interesting. They found a specific group of cells. They're called capillary endothelial cells, and they were really affected in people with those diseases. And these cells, they're the building blocks of the blood-brain barrier. And in disease brains, they showed this really distinct reactive signature, which basically means dysfunction and inflammation.
So these key barrier cells, they were basically stressed out and inflamed in people with these diseases. So where does TDP-43 fit into all of this?
Well, get this. The reactive capillary cells, the ones that were all stressed out, they had way lower levels of TDP-43 than healthy cells. And the researchers think that this loss of TDP-43, it could be a major reason why the barrier breaks down.
Okay, so less TDP-43, more messed up cells, weaker barrier. But how does losing one protein cause so many problems?
Think of TDP-43 like the conductor of an orchestra.
Okay.
It's orchestrating all these complex cellular processes. When the conductor disappears, well, the orchestra falls apart. The music is a mess. So in this case, when we lose TDP-43, it disrupts all these important pathways that are needed to keep the blood-brain barrier strong and healthy.
So it's not just is it there or not? It's about the chain reaction that happens when it's gone. Right. And that weakens the barrier.
Exactly. And the study even showed which specific pathways were affected. What's really interesting is they found problems in pathways that are all about blood flow and the forces of blood flow on blood vessels, which is kind of a new way to think about this.
Wait, blood flow and mechanical forces? I thought we were talking about proteins and cells. How does all that physical stuff come into play?
Okay, imagine a river that's flowing, nice and smooth. Now imagine that same river, but now there's all these dams and obstacles in the way. The flow gets all turbulent, right? And all that force puts stress on the riverbanks.
Oh, yeah, I get it. So you're saying that blood flow that's all disrupted can actually put stress on the blood vessels in our brain, which makes the blood-brain barrier more vulnerable.
That's what this research is suggesting, yeah. TDP-43 seems to be involved in regulating those forces, making sure the blood is flowing smoothly through the delicate vessels in the brain. When TDP-43 levels drop, that regulation goes out the window. And then the barrier gets stressed out from the turbulent blood flow.
Wow. So TDP-43, it's not just a conductor, it's a traffic controller too, keeping everything moving smoothly inside the blood vessels. This is complicated stuff. Can you just walk me through the purpose of the study again?
Sure. So the main goal was to figure out how endothelial cells, the ones that make up the blood-brain barrier, how they change as we age, and of course, in neurodegeneration. And they used a cool new technique that let them analyze what was happening inside specific types of cells in the brain, including those endothelial cells. It gave them a super detailed look at what was going on, and that's how they figured out how crucial TDP-43 is.
So it's like they had a tiny little camera zooming in on these cells and seeing how they act differently when there's a neurodegenerative disease going on. And this led them to TDP-43 as a possible cause of the blood-brain barrier breaking down. Sounds like a pretty big discovery.
Yeah, it's definitely a huge step forward. Now we're understanding how the blood-brain barrier gets messed up in these diseases. And by figuring out that TDP-43 is a potential target for therapies, this study opens up whole new possibilities for treatment. But, we do have to be cautious.
Oh, right. Before we get too excited, we should talk about what the study didn't tell us. What are some things to keep in mind about these findings?
Well, the findings are really exciting, but this was correlational research. It means they saw a link between TDP-43 being low and the blood-brain barrier not working right. But we need more studies to know for sure if one actually causes the other.
So we can't say for certain yet that losing TDP-43 directly causes the barrier to break down. There could be other things going on. It's a good reminder that science takes time, and we need more research to really understand what's happening. But this study still gives us really important information about the blood-brain barrier and how it's affected in neurodegenerative diseases.
So we were just talking about how researchers were able to find this specific group of cells called papillary endothelial cells, and how they were really affected in those with neurodegenerative diseases.
Yeah, it was really cool how they basically zoomed in on these individual cells and saw that they were like distressed and inflamed, especially in those with neurodegenerative diseases. And it all seems to go back to this TDP-43 protein, right?
Right. And what's even more interesting is that this study suggests that TDP-43 could play a huge role in how genes are expressed in these endothelial cells.
Wait, hold on, gene expression. I thought we were talking about proteins and the blood-brain barrier. How do genes even fit into all of this?
Okay, think of genes like blueprints. They have the instructions for building and maintaining our bodies, including those proteins like TDP-43. And these instructions, they're written in the language of DNA.
Got it. So to make a protein, the cell makes a copy of the gene's DNA instructions, but this time it's in the form of RNA. And then this RNA, it acts like a messenger. It takes those instructions to the part of the cell that builds the protein.
Exactly. So DNA is like the master plan, RNA is the messenger, and the protein is the final product.
Okay, I'm following. But you were saying something about regulating gene expression. What does that even mean?
So regulating gene expression, it's like having a volume knob on a stereo. It controls how much of a certain protein is being produced. Sometimes the cell needs a ton of a protein, and other times it only needs a little bit. And this level of production is super carefully controlled. And one of the things that does that controlling is, you guessed it, TDP-43.
So TDP-43 is not only like a conductor and a traffic controller, but it's also like a volume control knob for our genes. This protein does a lot. But I'm still not quite clear on how this all links back to the blood-brain barrier. What happens when TDP-43 levels go down? Does that mean certain genes get, like, turned up or down?
That's exactly what the researchers saw. When TDP-43 levels went down in those endothelial cells, they noticed this process. It's called RNA splicing. It started to go wrong.
RNA splicing. Okay, that sounds kind of intense, like cellular surgery or something. What exactly is happening there?
Think of it like this. RNA splicing, it's like editing a movie. You have a long film strip of all these scenes, and some scenes are really important to the story, and others, well, they're not needed. Maybe they even mess up the flow.
Okay, so RNA is the film strip, and splicing is the editing process. But what does this have to do with proteins?
So the RNA that's carrying the instructions to build a protein, it often has these extra bits of information in it. They have to be taken out, spliced out, before the protein can be put together correctly. This splicing process is super important. It makes sure the final protein actually works and does its job.
So it's like taking out the bloopers and the extra stuff from a movie to make a good final cut. And TDP-43 is part of this whole editing process.
Yep. TDP-43 makes sure the RNA is spliced the right way so the protein can be put together according to the original blueprint. But when there's not enough TDP-43, well, the editing gets messed up. And then you end up with these abnormal proteins that don't work right.
So it's like having a bad editor who accidentally cuts out important parts of the movie or leaves in things that shouldn't be there. The movie, or in this case the protein, just doesn't work. So how does all this bad editing affect the blood-brain barrier?
Well, the researchers found one of the genes that was messed up by TDP-43 being low was a gene called SORT1. This gene is in charge of making a protein called sortilin. And sortilin, it's really important for moving molecules across the blood-brain barrier.
Oh, so if SORT1 gets misspliced because there's not enough TDP-43, then we end up with less sortilin, meaning we have fewer of these transporter molecules that help regulate what gets into and out of the brain.
Right. It's like having fewer Border Patrol agents. It makes it easier for stuff that shouldn't be there to sneak across.
Okay, that makes sense. But is sortilin the only protein that gets affected by this messed up splicing?
They actually found that levels of a receptor that helps transport iron into cells and a transporter that helps move fats around were also affected by TDP-43 levels.
Iron and fats, what do those have to do with the blood-brain barrier?
Well, iron is really important for a lot of things cells do, like making energy and transporting oxygen. And fats, they're a big part of cell membranes, that outer layer of each cell. And they're also involved in the signaling pathways that help cells talk to each other.
So iron and fats are both basically essential to keep our cells healthy and working right. And TDP-43 could be involved in controlling how they're transported and used.
Exactly. The study found that when TDP-43 levels are low, the production and transport of these super important molecules gets disrupted. And that could mess up the ability of endothelial cells to do their job of maintaining the blood-brain barrier.
So it's like a shortage of really important building materials and fuel. It makes it harder for those endothelial cells to keep the barrier strong and secure.
Perfect analogy.
Yeah.
And when you combine this disruption with the inflammation and stress from messed up blood flow we talked about before, well, it creates this perfect storm that weakens the blood-brain barrier and makes it more vulnerable.
So it's basically a multi-pronged attack on the brain's defenses.
Okay.
From the outside, you have inflammation and turbulent blood flow just pounding on the barrier. And then from the inside, you have this faulty RNA splicing that's leading to shortages of key molecules and all these proteins that aren't working right. No wonder the barrier starts to break down under all that pressure.
Yeah, it's this really complex interaction of all these different things, and they all contribute to making this vital protective shield weaker. And figuring out these intricate mechanisms is super important if we want to develop good therapies to fight neurodegenerative diseases.
Okay, so we've seen that TDP-43 is crucial for keeping the blood-brain barrier healthy, and how losing it can mess up important cellular processes and weaken the barrier's defenses. But the big question is, can we do anything about it?
Well, that's what makes this research so exciting. Now that we've identified TDP-43 as a potential target for therapies, it opens up all these new possibilities for treatment.
So we're talking about drugs that could potentially protect or even bring back TDP-43 levels in those endothelial cells. Could this be the answer to preventing or at least slowing down these neurodegenerative diseases?
That's what we're hoping for. If we can stabilize TDP-43, we might be able to stop that whole cascade of events that leads to the blood-brain barrier breaking down and ultimately the development of these really tough conditions.
That would be amazing. But I'm sure there are some challenges, right? What are some of the hurdles that scientists need to overcome to make these kinds of therapies a reality?
Well, one huge challenge is actually getting those drugs across the blood-brain barrier itself. It's kind of ironic. We're trying to fix the barrier, but it's also what's blocking our treatments.
It's like we need a key to open the very door we're trying to fix. So how are scientists going to get past this obstacle?
Well, researchers are trying all sorts of strategies to get around the blood-brain barrier and deliver those drugs directly to the brain. One really promising way is to use nanoparticles. They're these tiny, tiny particles that can be designed to carry drugs and target specific cells.
Nanoparticles, those are like those little microscopic delivery trucks that can travel through the bloodstream and drop off their cargo right where it needs to go.
Exactly. And there are other cool approaches too, like focused ultrasound, which uses sound waves to create a temporary opening in the blood-brain barrier so drugs can slip through.
So it's like building a temporary bridge to get those treatments where they need to be. But it's not just about delivery, is it? We also need to make sure these drugs are safe and actually work.
Right. We don't want these therapies causing any unexpected side effects or harming other cells in the process. And of course, we need to make sure they actually work in people.
Makes sense. It's definitely a long and complicated road ahead, but the potential benefits are huge. If we can find a way to protect or restore TDP-43 in those endothelial cells, it could really change the lives of those facing these really difficult diseases.
And another interesting thing about this study is that it shows how important early intervention is. The researchers saw that changes in the blood-brain barrier were happening even before any obvious symptoms showed up.
So like early warning signs, that's huge. If we can figure out who's at risk early on, we might be able to do something before too much damage is done.
Exactly. Think of the possibilities for preventative therapies. If we could make the blood-brain barrier stronger before it starts to break down, we might be able to delay or even prevent these neurodegenerative diseases from developing.
Now, that would be amazing. But how would we even know who's at risk? Are there certain things like biomarkers that we could look for? Biomarkers, those are like those telltale molecules that can tell us if someone has a disease or a condition, right?
Right, and it's something researchers are working on right now. Some studies are looking into imaging techniques to try to see those early changes in the blood-brain barrier. Others are looking for markers in the blood or cerebrospinal fluid that might tell us if someone is at higher risk. And cerebrospinal fluid, that's the fluid that surrounds the brain and spinal cord, just to be clear.
So we're not quite there yet, but the future seems hopeful. Early detection and targeted therapies could completely change how we deal with these diseases. It's like having a crystal ball to see into the future of our brain health so we can be proactive and protect ourselves. So we've been diving deep into this groundbreaking research about TDP-43 and how it seems to be so crucial for that blood-brain barrier.
It's amazing how one protein can have such a big impact on keeping that barrier, that protects our brains, keeping it strong.
And it's pretty incredible that scientists are already looking into ways to use TDP-43 to develop new treatments for these neurodegenerative diseases.
It really shows how powerful scientific research is and how important it is to keep digging deeper to understand these really complex and, devastating diseases.
Speaking of complex, I know we've been talking a lot about TDP-43, but I'm guessing there's a whole team of players involved in keeping that blood-brain barrier healthy, right? What else helps keep it strong?
Oh yeah, for sure. The blood-brain barrier is this really dynamic and intricate system, and TDP-43 is just one part of it. Another really important part is the tight junction proteins.
Tight junctions, huh? That sounds like they're really good at keeping things out.
They are. Think of them like the mortar between the bricks of the blood-brain barrier. They make these super tight seals between the endothelial cells so nothing can sneak through.
So they're like the gatekeepers, making sure only the good stuff gets in.
That's a good way to put it. And just like with any well-guarded gate, strong, tight junctions are so important for keeping the barrier working right. But when they get weak or they're not working properly, the barrier can start to leak, and that lets all the bad stuff into the brain.
So it's not just about having enough TDP-43. We need those tight junctions in good shape, too.
Exactly. And researchers are trying to understand what makes those tight junctions go bad in neurodegenerative diseases. Some studies are looking at things like inflammation and oxidative stress.
We've talked about inflammation before, but what about oxidative stress? What's that?
Oxidative stress, well, it's basically when there's an imbalance between free radicals, those are like unstable molecules that can damage cells, and antioxidants, which are the good guys that neutralize them. When there's too much oxidative stress, it can really mess up cells, including the ones in the blood-brain barrier.
So it's like this battle going on inside our cells, and if the bad guys, the free radicals, win, the barrier gets weaker.
Exactly. And there's some evidence that oxidative stress might even contribute to both TDP-43 going haywire and those tight junctions breaking down. So it's kind of a vicious cycle.
Like a domino effect. One thing leads to another, and then the whole barrier falls apart.
Yeah, and that's why we need to think about addressing all of these things if we really want to protect the blood-brain barrier.
So it's not just about finding that one magic pill for TDP-43. We need to think about inflammation, oxidative stress, and all those other things that could be weakening the barrier.
Right, and that's where lifestyle comes in. Research suggests that things like exercising regularly, eating a healthy diet, and managing stress can actually help strengthen the blood-brain barrier.
Seriously. So what we do every day can impact the health of our blood-brain barrier.
It really can. Studies have shown that exercise, especially, can get more blood flowing to the brain. And that not only brings more oxygen and nutrients, but it also triggers the production of growth factors that help maintain and even repair the barrier.
So it's like giving our brains a workout, and we're making the barrier stronger at the same time.
And eating healthy foods with lots of antioxidants can help fight that oxidative stress and reduce inflammation, which also protects the barrier.
This is all making so much sense now. It's not just about targeting specific molecules with drugs. It's about taking care of our brains through our lifestyle choices, too.
Exactly. It's about thinking about the big picture when it comes to brain health.
This has been such an eye-opening deep dive. We've learned so much about the blood-brain barrier, why it's so important, and all the things that can affect it.
It has been a fascinating journey, and I hope our listeners feel like they can really take charge of their own brain health now.
Absolutely. By understanding the science and making brain-healthy choices, we can all work towards having stronger, healthier brains.
And don't forget, research is always moving forward. We're learning new things all the time, which gives us hope for even better treatments and ways to prevent these diseases in the future.
So stay curious, stay informed, and be proactive when it comes to your brain.
And never underestimate what you can do every day to make a real difference.
That's a perfect way to wrap things up. Thanks for joining us for this deep dive into the blood-brain barrier. Until next time, keep those brains healthy and keep those barriers strong.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.